continuation of my light reaction of my previous lecture, I'd like to discuss what is the pattern of distribution of different component of light reaction, how they are distributed in the thylakoid membrane. Now, we know the crucial role of the thylakoid membrane in the light dependent reaction of photosynthesis. Basically, it consists of two main regions, the appressed region and non-appressed region. So, you see that in the figure, appressed region is in the light color, whereas in the non-appressed region appears dark in color. And it is very important to mention here that photosystem 2 units are located almost exclusively in the appressed region, while photosystem 1 and ATP synthase are located in non-appressed regions. The cytochrome B6F complex is uniformly distributed throughout the membrane system. Now, I will little bit elaborate what this appressed regions contains. Appressed region of the thylakoid membrane refer to areas where thylakoid membranes from adjacent grana stacks come into close contact, or you can say tightly packed together in stacked form. These regions are characterized by tight stacking of the grana, resulting in a very you see that compact and organized structure. The close basically, what is the motive of this compaction of in the appressed region? It makes close proximity of the thylakoid membrane in the regions and facilitate efficient transfer of electrons between photosystem and enhances the efficiency of ATP and ADPH synthesis during photosynthesis. And these appressed regions are typically associated with high photosynthetic activity and are found in the regions of thylakoid membrane exposed to intense light. Now, what are the non-appressed regions? The non-appressed regions of the thylakoid membrane refer to areas where thylakoid membranes are not tightly packed together and exhibit a sort of more dispersed arrangement. These non-appressed regions are basically characterized by a loose arrangement of thylakoid membranes, resulting in a less compact structure compared to appressed region. The non-appressed regions provide a larger surface area for diffusion of ions, metabolites and proteins within the thylakoid lumen, facilitating various sorts of biochemical processes associated with photosynthesis. So, you see these appressed and non-appressed region individually play a very pivotal role in light reaction. Now, question arises with the movement of electron, transfer of electrons and movement of proton across the membrane and inside the membrane. What is the significance of all these movements of protons and electrons to different carriers? So, in this lecture I will discuss how ATP is synthesized during photosynthesis. As you know that once electron leaves the photosystem, it is basically transferred to a series of molecules, first to plastoquinone, then to a cytochrome complex and finally to plastocyan. During this electron transport chain, the electron moves from higher to lower energy level and thereby releasing energy. So, what happens? Some of this energy is used to pump protons from stroma into the thylakoid interior. The transfer of protons and release of protons from splitting of water molecules basically creates a proton gradient across thylakoid membrane, Now this gradient is utilized to synthesize ATP. So during light reactions of photosynthesis, protons accumulate in the thylakoid lumen and establishing a proton gradient that activates ATP synthase, which is an enzyme complex embedded in the thylakoid membrane, just I have shown in my previous lecture. Now protons then move through the ATP synthase from the thylakoid lumen to the stroma, driving a rotational mechanism within ATP synthase that converts the proton gradient's potential energy into mechanical energy. This mechanical energy now facilitates the phosphorylation of ADP and inorganic phosphate and synthesize ATP. Most importantly, this ATP synthesis process is generally coupled to the electron transport chain occurring in the thylakoid membrane, and this further demonstrates the interconnected nature of energy production during photosynthesis. Basically, in oxygenic photosynthetic organisms, water molecule acts as an electron donor. Photooxidation of two water molecules generally yields 4 electrons, 4 protons and 1 molecular oxygen. As we have discussed, these electrons are transported down the electron transport chain, and some of the energy released is basically used to pump proton across the thylakoid membrane from the stroma of the chloroplast to the thylakoid lumen, and this generates a sort of what? Electrochemical proton gradient. Now this electrochemical proton gradient generates a proton motive force. Hence, what happens in total, the absorption of 8 photons by both photosystem, that is photosystem 2 and 1, 4 photons each, and photooxidation of water molecules yields how much? 4 protons and 1 molecule of oxygen into the thylakoid lumen. And it is generally estimated that transport of 4 electrons through cytochrome B6F complex results in the translocation of 8 protons from the stroma to the thylakoid lumen. Hence, you see that total 12 protons are released into the lumen. The accumulating protons in the thylakoid lumen pass back across the thylakoid membrane to the stroma through ATP synthase, and this ATP synthase is also known as coupling factor 0 and CF1. ATP synthase, basically what it does? It catalyzes ATP synthesis as proton flow back along the electrochemical proton gradient. And the chloroplast ATP synthase produces 1 ATP per every 3 protons it transports out of the thylakoid lumen. So basically, non-cyclic electron transport therefore results in the production of approximately 4 molecules of ATP per molecules of oxygen evolved. So now the proton gradient created by the movement of electron is essential for the synthesis of ATP, and it is very special to mention chloroplast ATP synthase harnesses the energy of proton gradient to catalyze the synthesis of ATP from adenosine diphosphate and inorganic phosphate. This process is known as chemiosmosis, which involves basically involves the flow of protons through the ATP synthase complex, driving the rotation of a rotor subunit within the complex. This rotation leads to conformational change in ATP synthase complex that enables the synthesis of ATP from ADP and inorganic phosphate. So it could be summarized that ATP is basically generated during the light reactions. Protons accumulate inside the thylakoid interior, creating a sort of concentration gradient. These protons now diffuse back into the stroma and their passage occurs only through the enzyme ATP synthase. ATP synthase utilizes the flow of protons to convert ADP and inorganic phosphate into ATP through a process called chemiosmosis. So, now you must be clear that how ATP is synthesized in chloroplast. Now, as you know that electron moves proton cross from thylakoid lumen into the stroma. So, there are certain inhibitors which inhibit photosystem 2 and photosystem 1 activity. Now, these inhibitors I will discuss. There are two inhibitors role namely DCMU and paraquat. Now, what is DCMU? DCMU is 3,3,4-dichlorophenyl 1,1 dimethylurea. This is also known as diuron. Is a sort of herbicide that disrupt photosynthetic oxygen production by targeting photosystem 2. You see in the figure that it functions generally by competing with plastoquinone for QB binding site of photosystem 2, thereby block the flow of electron from photosystem 2 to cytochrome B6F complex, and basically it halts the transfer of electrons within the photosynthetic electron transport chain, ultimately inhibiting the production of oxygen. On the other hand, you see how paraquat blocks the transfer of electron. Paraquat is basically a kind of herbicide that acts by interfering with photosynthetic electron flow and generally operates by intercepting electrons between the ferredoxin acceptor and NADP, preventing their transfer from production of NADP to NADPH. Paraquat generally redirects these intercepted electrons to reduce oxygen molecules, forming superoxide radicals, and this superoxide radical is highly reactive free radical that can cause damage by reacting non-specifically with various molecules within the chloroplast. So, this indiscriminate activity generally leads to the rapid impairment of chloroplast activity, contributing the herbicidal effects of paraquat. Now come to we have gone through in detail the non-cyclic photophosphorylation. Now, what is cyclic photophosphorylation? What happens in certain cases, electrons deviate from the typical path and repeatedly cycle through photosystem 1 instead of ending up in NADPH. This phenomenon is known as cyclic photophosphorylation. What happens after leaving PS1, these flowing electrons return to either the cytochrome complex or plastoquinone in the initial part of the electron transport chain. Subsequently, electrons flow down to the chain to PS1 as usual, facilitating proton pumping and ATP production along the way. However, what happens in the cyclic pathway? Electrons bypass NADP reductase. So, NADPH is not generated in cyclic photophosphorylation. The question here that comes in our mind, why does a cyclic pathway exist? In certain situations, chloroplast can switch from linear to cyclic electron flow when there is an imbalance in the ratio of NADPH to NADP due to insufficient availability of NADP to accept electrons. However, what happens? Cyclic electron flow is often found in photosynthetic cell types that particularly have high ATP demand, such as bundle sheath cells responsible for C4 photosynthesis, which involves synthesizing sugars. Additionally, cyclic electron flow could serve as a photoprotective function by preventing excessive light from harming photosystem proteins and facilitating the repair of light-induced damage. Now, question arises, what are the basic differences between photosystem 2 and photosystem 1? So, this slide shows the comparative account of both photosystem, and the main differentiating point between photosystem 2 and photosystem 1, or you can say non-cyclic photophosphorylation and cyclic photophosphorylation. You see that both PS2 and PS1 are involved in non-cyclic photophosphorylation, whereas in cyclic phosphorylation, photosystem first is only involved, and location of these photosystem also differ widely, and you see that photosystem 2 lies in the appress region, whereas photosystem 1 lies in the non-appress region. And the most significant point in the absorption of light, that PS2 absorbs light most effectively at 680 nanometer, whereas PS1 absorbs light most efficiently at 700 nanometer. So, PS2 has different reaction center, PS1 has different reaction center, and PS2 is basically involved in non-cyclic photophosphorylation, whereas PS1 is involved in non-cyclic and cyclic phosphorylation. In PS2, you see that photolysis of water takes place, whereas in PS1 there is no photolysis of water. And in PS2, in non-cyclic photophosphorylation, oxygen is produced, whereas in cyclic phosphorylation, you see there is no production of oxygen. And in photosystem 2, there is a multi-subunit complex composed of about how much? 25 to 30 subunits, whereas a smaller complex with 15 subunits are present in photosystem 1. Now, in this lecture, we have learned the basic outline of light reaction of photosynthesis, and in my next lecture, the detailed mechanism of next phase, that is photochemical reaction or dark reactions, I am likely to discuss. Thank you very much.